Okay, welcome to podcast 41, and we've been talking about evolution and natural selection. In this podcast, we are going to look at how we could actually study evolution, how we can look at a population and determine if it's evolving or not, and what exactly that would mean if we could see it. Um, this is the study of population genetics, and we let's just dive into what this means. Okay, um, fundamentally, populations are the smallest units we have that evolve. Um, and what we mean by evolve is that the occurrence of a trait, or more specifically a gene that codes for a specific trait, changes over time. That gene gets more frequent or less frequent in a population of critters, I don't know, squirrels, over time, over years. If a trait is favorable to an organism, to a population of organisms, we would expect that trait to get increasingly common according to natural selection. Um, if a trait is unfavorable, we would expect it to get less common over time. So this gene would get more common or less common over time. Populations evolve. Natural selection acts on individuals in a population. If an organism pops up with a trait that's not favorable, then natural selection might act against it. In other words, it wouldn't survive. It wouldn't be able to pass that trait on to its offspring. But an individual does not evolve. If I had a child tomorrow that had a really great trait, that doesn't mean the entire human population has evolved. It means my kid got a great trait. If over thousands of years, millions of years, that trait becomes increasingly common in the population, then we might say that some sort of evolution is occurring. So, individuals do not evolve. Okay? Individuals either survive or they don't survive. That's natural selection acting on the individual. Um, Individuals do not evolve. Individuals are selected for or selected against. They are allowed to survive or not survive. They are allowed to pass on their genes to their offspring or not. But it is populations as a group that evolves, because what we see is a particular trait getting increasingly common or more uncommon in that population over time. Okay? There are two types of evolution that we often talk about. One is this macroevolution, which is essentially what we think of when we think of evolutionary change. We think of going from one species to a different species. Big change that we can see in the fossil record, for example. Big changes over time. Evolution on a grand scale, as this says here. Let me get my pen. There we go. Evolution on the grand scale of things, going from bacteria to multicellular animals to dinosaurs to us. That's big evolutionary change. Okay? Microevolution, on the other hand, is the other type of evolution we will talk about. And this is looking at the little tiny changes that might lead to macroevolution. Change in the genetic makeup of a population from one generation to the next. Is this gene that codes for this trait becoming more common as we go from one generation to the next, or less common? We humans cannot observe macroevolution happening directly. it's huge change. If we look at macro, these kinds of changes over time, those are grand changes, and it takes too long for us as humans. We can't watch those kinds of things happen. However, we can watch microevolutionary change, and we do watch it. Much of our biological science is based on, particularly in medicine, is based on watching it. The whole field of antibiotics is governed on watching these small microevolutionary changes happen in organisms. The peppered moths are a classic example of just microevolutionary change. When tree trunks in a certain part of England, I believe, where these were studied, or Great Britain, were gray with lichen, the more common phenotype of moth to observe is the gray peppered moth that would blend in very well in this tree trunk. Black forms would pop up because the gene existed in the population, but they'd be so visible that they'd get quickly eaten. Natural selection would select against them. They wouldn't get to pass that trait on, so they never got very common. However, when environmental conditions changed such that the tree trunks became black, like we see here, due to environmental pollutants, what we see then is that if a black moth popped up, all of a sudden his chances of survival increased, and so we saw the black moths getting more and more common in the population, whereas these lighter colored moths became less common. Was that macroevolution? Did we get a new species of moth? No, we didn't. We saw a gene in the population becoming more common or less common depending on its environment. But the idea is that these small changes could accumulate. Other small changes might accumulate in this moth population over thousands of years that might actually lead to a group of moths being different enough from its parent moth population that they would be considered a new species. Okay? So the idea is that microevolution is what leads to macroevolution. Microevolution are these tiny changes, and if you accumulate such tiny changes over many, many, many years, you lead to macroevolution. So microevolution is what leads to macroevolution. It is what we can observe most directly, although certainly we can see the effects of macroevolution. If we look at the fossil record, we can certainly see that, but we can't observe it directly. Now, as scientists, we seek to be able to measure things happening. Can we monitor it? Can we see it? Can evolutionary change be monitored as it occurs? It can. Scientists do it all the time. But we are talking about microevolutionary change. How do we measure that? It is as simple as measuring how often we see a trait occurring in a population over time. We pick out a particular trait of interest. Is this trait getting more common over several generations or less common? If it is getting more or less common, then some kind of evolution is occurring. So we are measuring how the numbers of different alleles in a population and different genotypes in a population change over time. Okay, so let's get some basic terms down. First of all, we must know what a population is, because that's what's evolving. A group of organisms of the same species occupying a certain geographic area and sharing a common gene pool. In other words, they are interbreeding with each other. So, what's a gene pool? The total of all the genes of all the individuals in a population. So if our gene is big A and little a, how many of these are present in the population among all the individuals? We might have I don't know, 30 individuals in our population that are big A, big A. We may have 20 that are little a, little a. If that's the case, then we've got 60 big A alleles. We have 40 little a alleles in this, population of, I don't know, deer. All those little specks are deer. And so we've got 50 deer, 30 are big A, 20 are little a. Then we would have 60 big A's, 40 little a's. That's the gene pool. That's what they have to work with. Okay? Genetic structure is simply describing how the population looks genetically. How many of a particular allele is in the population, how many of a particular genotype is in a population. Let's talk about this. Genetic structure tells us the allele and genotype frequencies in a given population. So let's talk about what an allele frequency is. Allele frequency is just a number that tells us how often an allele is in a population. So let's say we have a population of deer and there's some trait, big A. Okay, so let me go back. There's some trait, big A. Let's say it's brown fur. Okay, and there's some trait, little a. Let's say that's white fur. Okay, and I've got, I don't know, 30 individuals that are big A, and I've got 20 individuals that are little a, okay? If I want to know how many big A alleles are in this population, well, if I know I have 30 critters that are big A, then I know that each individual has one big A, two big A alleles in their genotype, and I know I've got 30 critters, so 30 times 2 is 60, and that would be my allele frequency. Well, actually, I lied. In the population, I have 60 of these big A alleles. My frequency would be this number divided by my total number of alleles in the population. So what is my total number of alleles? Well, there's 2 big A's times 30, so that's 60, plus 2 little a alleles times 20 is 40, so 100 alleles. So 60 divided by 100 is 0.6, and that would be my allele frequency for the big A allele. My allele frequency for the little a allele would be 40 divided by 100, which would be 0.4. So an allele frequency is telling us how often an allele occurs in a population. So if my allele frequency for big A is 0.6, that means 60% of the time, if I had all of the alleles for my population in an allele bag, a grab bag, that means 60% of the time I would reach in and pull out a big A allele. That is what we mean by allele frequency. How often is that big A allele occurring in the population? Okay, how many big A versus little a in the population? Genotype frequency is the exact same thing, only instead of just alleles, I'm talking about full genotypes. I'm talking about individuals. How many big A individuals are there in my population? How many little a individuals are there in my population? How many times would I pull out a deer and have that deer be big A? And that is what we mean. So how many big A versus big A versus little a, okay? So, in order to measure evolutionary change, then what we assume if evolution is occurring with regard to a trait, fur color, whatever, we occur that the number of alleles, the big A's versus the little a's, or the genotypes would not remain the same over time. If evolution is occurring with regard to that trait of fur color, then the frequency of the big A allele for brown fur would get more common if that was the favored trait, and the frequency for little a, the white fur, would get less common over time. So if we want to determine if evolutionary change is occurring in a population, we just count how many alleles, how many genotypes of each type we are seeing in the population. If it is staying the same, then no evolution is occurring. If it's not, then we assume some sort of microevolutionary change is occurring. Okay? So, in order to measure evolutionary change, then, microevolutionary change, we have to be able to measure how many genes, how many alleles at a particular locus for whatever trait we're interested in are present in a population at a given point in time. Then we come back and look later and say, okay, is it the same or not? Okay? If the numbers are different, if I have more big A's at a later time than I do have when I first started testing, then we must have to consider that some kind of microevolutionary change is occurring. Okay? Hardy and Weinberg are two scientists that basically said this. They said, if the genetic structure, if the allele frequencies change over time, then, or genotype frequencies change over time, then some kind of evolutionary natural selection pressure must be acting on the population. Because they also said, if nothing is acting on the population, no evolutionary pressures, no natural selection, nothing is acting on it but sex and fertilization, meiosis and fertilization, that the population, the genotype and allele frequencies should just remain constant, the same over time. If no evolution is occurring, then that population should genotypically and in terms of allele frequency remain the same. Okay? They proved mathematically that without natural selection, in other words, with only meiosis and only sexual reproduction, here is my little population of deer. Okay, here they are, all happy in the woods, and the only thing that's acting on them is their own meiosis, creation of sex cells, and their own sexual reproduction within their population with each other. There are no deer coming in, there's no deer leaving, there's nothing, there's no natural selection favoring one over the other. If that is the case, then no evolutionary change should occur. They can shuffle their alleles as many times as they want within their population via meiosis and sexual reproduction, and nothing should happen to the allele or genotype frequencies. Okay? That's what they showed us. Now, let's think about what that means for just a second. So, if no evolution is to occur, they said that, first of all, we do have to have a very, very large population. We can't just have 2 deer in our population and have no evolution. It has to be a large population. We'll talk about why later, but the population has to be large. We can't allow any deer in or out. No gene flow in or out, no mutations. Now, we can't have any new genes just pop up spontaneously. We can't have gray fur pop up in our brown and white discussion. Mating has to be completely random among these deer. So here's my deer. They are not allowed. This deer here cannot say, ooh, that deer over there is a cutie, I want to mate with him. They can't do that. And above all, no natural selection. One deer is not allowed to be favored by the environment over another. But they said if all these things were in place and only meiosis, I can't spell, and fertilization were the things acting on them, the number of big A's and little a's in this population would stay the same generation after generation after generation. Okay? Now, we all know that there are very few populations on planet Earth that are going to meet these criteria. So why even discuss it? Why is this of value? Why is what they gave us important? Because what they gave us is essentially our null hypothesis with regard to evolution. Since mathematically they showed us that if you have this population in this bubble, meiosis and sex can happen as much as you want in that bubble, but after generation after generation after generation, the allele and genotype frequencies will stay the same. That is your control treatment right there. You have a mathematical control treatment such that we know that if we actually go out in the wild and measure the genotype and allele frequencies in a population at point A in time and then come back years later at point B, and they aren't, the frequencies of the alleles and genotypes are not the same, then we know that some kind of evolution has occurred. Okay? So it is a useful model to measure if evolutionary forces are in fact acting on a population. Okay? It doesn't necessarily tell us what those evolutionary forces are doing or what's causing it, but it does tell us that evolutionary forces are acting on a population. So let's try and look at this. The Hardy-Weinberg theorem is an equation, and we are going to work with allele and genotype frequencies, and I'll try and show you how we do that. We are looking at a population of kitties. Let's assume we have two alleles here. We'll call them big B is black fur and little b will be white fur. Okay? In the Hardy-Weinberg theorem, the frequency of the dominant allele is always written as P. So in this case, the frequency, how often big B occurs in the population, is written as P. Okay? The frequency of the recessive allele is written as Q. Okay? How often the little b allele occurs in the population. Okay? And these frequencies add up to 1. So the frequency of the big allele plus the frequency of the small allele in the population always equals 1 or 100%. Okay? Now, if we want to look at individuals, how many big B individuals, how many big B, little b individuals are there genotypically? We can, if we know P or Q, we can figure this out. The frequency of the big B homozygous dominant is simply P times P, or P squared. The frequency of that big B allele times the frequency of the big B allele is P squared. Okay? Same goes for the recessive allele. Q squared it gives us the recessive genotype, how many actually white cats we are going to have, for example. And because there's two ways to get the heterozygote, remember, you can get big B from mom and little b from dad, or you can get little b from mom and big B from dad. You multiply P times Q, the frequency of big B, times the frequency of little b, but you also have to account for the fact that there's two ways to get that, so you multiply by 2. So 2 times P times Q is going to give you the frequency of your heterozygotes. Alright? Now the frequencies of all those individuals again have to add up to 1. So there's an equation that ought to look pretty familiar to you. You no doubt have seen this equation before. P squared plus 2PQ equals Q squared equals 1 or 100%. Okay? This is the Hardy-Weinberg equation. Alright? So let's get these straight. This is our equation for just alleles, just the frequency of big B or little b in the population, okay, and our equation for actual genotypes, individuals, okay, and that's the frequency for how many of these big B black cats we'd see. This is the frequency for for how many of the little b white cats, and then you've got your heterozygotes as 2pq. Alright, so let's see how this works. If you have a population of cats, you have 84 black and 16 white. Now, if you look at these black cats phenotypically, you can't tell which ones are big, which ones are big, little. But by golly, if you look at these white cats, you certainly do know their genotype. You know they have to be little b. But how can we figure out how many of each genotype we have? So, we already know how many of these white cats we have, and we know their genotype. So we can determine q squared, which is the frequency of the little b's, by simply taking 16. We've got 84 plus 16 equals 100. So 16 white cats divided by 100 cats total gives us 0.16. So that is the value for q squared. Now once you've got the value for q squared, which is always easy in most of these problems because you can see the homozygous recessive individual. You know how many of them you have. Q squared's easy. Once you've got q squared, you can get all the rest of the values. How can you get q? The first thing you do is take the square root. If you take q squared, the square root of q squared, it gives you q, so the square root of 0.16 gives you 0.4. So that is the value of q. Well, if you've got q, since we know that p plus q equals 1, we also know that 1 minus q equals p, so 1 minus 0.4 is 0.6. So we know that p equals 0.6. Once we know that, then we know everything else. If this population is in Hardy-Weinberg equilibrium, that's a big if because we know most populations aren't, but if it's in Hardy-Weinberg equilibrium, then p squared, 0.6 times 0.6 is 0.36. Q squared we've already got is 0.16, and 2pq, 2 times 0.4 times 0.6 is 0.48. Now, that's what this population ought to look like if it's in Hardy-Weinberg equilibrium. 48% of the time, if we draw a cat out of this population, it ought to be big B, little b. Now, if we assume Hardy-Weinberg equilibrium, that's kind of our null hypothesis. We would expect this proportion of cats. But what if we go into our cat population and really study it, and we find out that our cat population, once we sample it, is actually this? Well, then we know they're not in Hardy-Weinberg equilibrium and something must be going on. What is going on? How would we explain that? Well, clearly, who is natural selection acting against in this population? Who is not in the numbers that we would expect them to be? Clearly, 0.10 is much lower than 0.48, so something is acting against these heterozygotes. What? Who knows? That's not answered by Hardy-Weinberg, but something is acting against them and making them lower and favoring these homozygous dominant individuals. They are much more numerous in the population than what we would expect if Hardy-Weinberg were the case. So, suppose, however, we looked at the population and we saw this kind of result. Now it appears that something is favoring the heterozygotes. It's now larger than what we would have expected if Hardy-Weinberg equilibrium were the case. And these homozygotes are being reduced compared to what we would expect. So this is how Hardy-Weinberg, the equation can be used as kind of a standard, a control treatment for us to look at a population and say, okay, do these match Hardy-Weinberg? No, then evolution must be occurring. Why? Well, then that's the problem. It's a little bit harder to figure out. Okay? So, the Hardy-Weinberg equation is a tool to determine if the frequencies of alleles in a gene pool, if we know certain genotypes and vice versa, and it is useful because we can figure out what the frequencies should be if the population is in Hardy-Weinberg equilibrium. Okay? All right, let's do a practice problem. In human populations with regards to certain traits, evolution is happening slow enough that we can actually use Hardy-Weinberg to just help us to determine allele and genotype frequencies roughly for the entire human population. So let's use it in this way. PKU is a recessive disorder in humans. It can be treated, but you're all tested for it at birth. When you were born, you've got a little heel prick and a blood test to determine if you had PKU. And the reason being is that PKU is just a disorder in which you cannot process an amino acid properly, and if you have PKU and you ingest foods containing this amino acid, you can have very serious consequences. Mental retardation and things can happen, but all of that can be avoided if you just don't eat any foods that contain that particular amino acid. So everyone is tested at birth for that, so that immediately dietary restrictions can come into play, and the person will be fine. The disorder does occur at a rate of 1 in 10,000. Okay? So if I asked you what's the frequency of carriers in the human population of this disorder, and I told you just assume that Hardy-Weinberg is a place, assume that no evolution is going on, can you figure out what the frequency of carriers for PKU is? It's a recessive disorder, 1 in 10,000 births. So what number have you been given? You have been given 1 in 10,000 has PKU. So you have been given Q squared, the frequency of the recessive individuals. Okay? So if you want to know how many little a alleles there are, or sorry, let's do this. I think we'll work this in class. Let's just step back and we'll work this in class. Okay, so in summary, Hardy-Weinberg can tell us what a population that is not evolving ought to look like, so we have a standard of comparison, and we can use it as a reasonably accurate tool to get the genetic structure of a slowly evolving population. Okay? Again, there's no populations on Earth that aren't evolving, but if it's evolving slowly, we kind of say, okay, this is probably more or less the genetic structure of this population at this point in time by using Hardy-Weinberg. All right, we're going to stop there, and I will catch you in the next podcast. Take care.